A huge thank you to Defeat Duchenne Canada, and thank you to all of you for sticking around to the end of the session. So much appreciated. We will hopefully be lively for the last 10 minutes here, and I'll try to keep to time as well. But really wanted to introduce you all to the program that you may not know too much about. Some of you do, some of you don't. But we want to go through an overview of what we are doing for Duchenne right now, which is our SGT-003 program, which is a microdystrophin gene therapy. But we consider this a next generation microdystrophin gene therapy, and it's really because of key aspects that we've updated along the way. We are a public company, so I do have that forward-looking statement slide. Please don't read it. And for those of you who don't know Solid, just a very quick intro to us. We were founded by those who were impacted by Duchenne. Our founders have a son with Duchenne muscular dystrophy, and so it's been our core focus since the beginning of the company. It's what I did my PhD on. It's what we continue to really focus on. More recently, we have expanded into now working on additional neuromuscular and cardiac indications, with Duchenne still really being the main focus, and this program really being what we're really looking forward to seeing what comes from the next steps and the changes that we've made. So I think we all really understand Duchenne, and that ultimately when we have to develop a treatment for Duchenne, we need to be able to actually address the root cause. Here there's a lot of information on this slide, but the reason that we're working on a microdystrophin gene therapy is because we know that we need to restore that muscle integrity that's lost when the protein itself is lost. And so with that really comes the identification of pieces that are necessary, not just for skeletal muscle, but also for cardiac muscle, really to be able to have the potential most optimal impact when we can replace what's missing here. And that really goes to the identification of the different components that went into SGT-003. The first, and I'll actually give a great call out to Hernan Gonarowski earlier for doing an excellent overview of gene therapy and really each of these components and why they matter so much. The first here is that transgene. And so there was discussion on not just being able to have an amount of microdystrophin that's restored, but actually you need to have a quality protein that's restored. And so that's the reason for why we chose the microdystrophin that's in SGT-003. On the next slide, we'll do a little bit more of a dive into it, but really it's so that we could be able to restore more functional domains of a full-length dystrophin. As was mentioned, we need to make choices because it's such a large protein and what we're using as our capsid can't fit the entire thing. So we want to have potentially what can be the most optimal benefit. In the center here is really the core of what is the program, and it's this capsid. So what was spoken about earlier was the difference between a capsid that is naturally occurring, these AAV8s and 9s and 2s, those that can get to muscle, versus those that are actually more muscle tropic, that are next generation capsids where you can actually modify the capsid surface so you can better target muscle. And that is what we did for this program. If anyone knows Solid from the past, we had an SGT-001 program that used an AAV9 vector. It didn't get the expression levels we wanted. We really wanted to go back to the drawing board, understand what can you change from a meaningful side, and that's why the capsid was replaced to actually use what we call AAV-SLB101, which is an additional muscle tropic capsid that gets more to the skeletal muscle, more to the heart on multiple fold higher levels, and half of the amount to the liver. So more where we want it to go and less where we don't need it to go and where it could be actually a safety concern for it to go. And so those were emphasized earlier, and they really go to the foundation of this program. The last component here is the manufacturing process. And so as we make vector and we make large amounts of it, we need to make sure that it's pure, because we are ultimately still delivering a large amount of virus to our patients. And with that comes safety concerns that we need to try to mitigate as much as possible. When we can focus on purity, it gives us the ability to actually dose more accurately and give less of an ultimately high total viral load that comes with that impurity process that can happen when you scale up in manufacturing. And so we talked about how we chose our microdystrophin. I won't go through what was reviewed earlier on the capacity of an AAV, but looking at that full-length dystrophin, we can see that there are pieces that are absolutely critically essential, and they are really the structural components of a dystrophin, where we have to link the intracellular membrane, the actin cytoskeleton, to the extracellular matrix, and that's the core function, again, of a dystrophin. However, when we look at the central rod domain, there's a lot of redundant function there, but there is also some unique function. The reason for our choice of including the R16, R17 repeats in our construct is because that's actually able to restore nNOS to the membrane. The reason that matters is because nNOS functions where it's localized. And so if it's intracellular, if it's not actually bound to the membrane, it's not going to result in the blood flow increases that are required. So when it's at the membrane, it's able to actually function. And when you have better structure, you also need to have more energy that gets to your muscle. Otherwise, that can result in damage. And so that's really the reason for why we put these two pieces together to be able to select this microdystrophin for our SGT-003 program. And so overall, the two studies that we're currently running and beginning to run are how we're thinking about our overall clinical development. Right now in the clinic is the INSPIRE Duchenne study. This is a phase 1/2 study. That was our first in-human study that just started dosing at the middle of last year. So we're very happy to say that we began that study with an FDA IND approval in the US. We quickly moved into Canada, and it's been amazing working with SickKids, Hernan Gronarowski, and Anna Stosik and the entire team over there. They've been amazing partners, and it's really just exciting because we've had really close links as Solid to SickKids and Ronnie Cohn and others for many, many years. So we've always wanted to move in this direction, and it's been great to be able to do so. We expanded that out into Europe as well. So we have a site in Italy as well as in the UK. However, as we're thinking about the overall development of this program, we know that as we go into other regulatory regions, especially in Canada, in Europe, we need to do more than just an open label study because that's really what's required. That session that was this morning was very, very complex in thinking through what's the approval process and what's the reimbursement process. And unfortunately, one of the major things when it comes to both of those aspects is having placebo-controlled data from a functional efficacy study. And so it still remains a requirement, and it's why we don't want to wait until a later point to be able to get that moving, but we're actually working on initiating that now. You can see here that it will be focused on patients who are going to be ages 7 to 11 so that we can limit the variability a bit in this overall population to be able to detect a treatment effect. And it will be ex-US. So we're working very hard to get that into Canada especially and continue working with Canada. I'll go through a few of the high-level details of each one of these studies just for a good informational purpose. And as I mentioned, INSPIRE Duchenne is our ongoing study that does have current data as well. So I mentioned this is currently running in the United States, Canada, the UK, and Italy. We have 14 active clinical trial sites, so we're very excited about that. The beginning of the study was 2 sites. We were able to basically get a good understanding of what are we seeing from our product from a safety, an initial biomarker perspective, and from there we quickly ramped up and expanded. And so this year has really been on that, and we'll continue to be dosing in this study as we move forward. This enrollment is a bit more wide in age range compared to what I mentioned about our phase 3 study. So here what we want to do is really learn as much as possible. We want to dose patients who are going to be less than 12 years of age, and we just recently opened a cohort that's actually under 4 years of age. And so we've been really excited to move in that direction to learn more, and especially to be able to enroll as many patients as possible and to understand what is the safety profile of SGT-003 and what is the overall biomarker profile, and as we move forward, looking at more of the functional efficacy outcomes as well. Now, in the first 15 participants, this is our last data cut that we had, especially looking at safety, we were really encouraged to see that really what we were seeing was a very similar profile to most gene therapies, where we had nausea, vomiting, reduced platelet counts that were occurring, but these were more of the mild symptoms that we would expect and hope to see within the study. What we weren't seeing were drug-induced liver injury across any of the patients who were enrolled, and that's been a very encouraging thing that we have monitored very closely, especially based on more of the recent updates over the year. And now going into some of the data that we've generated, when we look at muscle biopsies after dosing, what we can look to see is, are we expressing our protein? And you can see the baseline is on the left side and the day 90 biopsies are on the right. And we can see that we are expressing microdystrophin and it's localizing to the muscle membrane. So it's showing that it is molecularly functional in actually getting to the levels and distributing throughout the muscle to points that we would want to see. We then have an impact on the dystrophin-associated protein complex, where we can see that beta-sarcoglycan, which is a critical complex member in that it actually restores additional proteins to the complex and will assist in being able to maintain that overall muscle integrity. We see that that is restored in our microdystrophin-positive fibers as well. And then unique to SGT-003, we also result in the restoration of nNOS activity in these microdystrophin-positive fibers. So with that, we can look at more biomarkers, and we look at things really across the body that we can see both in the circulation and then in muscles. And so because this is a muscle integrity analysis, we really want to focus on things that would give us an idea of what's happening in the muscle, both from a muscle pathology as well as from a leaky membrane standpoint. And so many of these things you may have heard of before, and we can see that things like serum CK is going to be significantly downregulated. We actually see that we have about 50 to 60% reductions at our day 90 time point. And those are consistent with other markers as well that are also showing that they are not being leaked from the membrane. So we have overall muscle integrity that's restored, as well as things like EMHC, where we see that we have less levels of active degeneration and regeneration. So actually stability of the muscle, which is extremely important. And then lastly, moving over into what our phase 3 study is, which we are currently working on getting started. As an overall overview, what I mentioned is that this is going to be a placebo-controlled study. It is called Impact Duchenne. So we started with Inspire, and we're moving to Impact to really see what is that impact of our therapy on the overall functional capacity of the patients. This will be a one-to-one randomized study where patients will get a single dose of SGT-003 or placebo. At 18 months post-dosing, we will have the ability to then have all of those who are on placebo receive SGT-003 if they remain eligible from a safety perspective only. So that's ultimately what we will monitor at that point. I'll emphasize that the reason that we focus on 18 months is really based on learning what we have now come to know from the field, in that it takes time to be able to appreciate a change. We always want to limit these study sizes and lengths as much as possible, but the worst thing that we can do for a study is actually to design it in a way that it's not going to be successful. And so if we look back at things like the givinostat study, which we'll always reference, if you actually look at the 12-month endpoint, it would not have been successful if they did not go out longer to 18 months. And so while it does take longer to get to that point, it's that critical importance to be able to actually get a drug to as many patients as possible to get that overall approval. This study will be focused on 7 to 11-year-olds, and we are looking at an ambulatory endpoint for it. We mentioned some of our exclusions, and this is common between each of our studies in terms of the actual mutation exclusions, and they're deletions only that are excluded, and these are exons 1 to 11 and 42 to 45. The reason, again, being the ability of these to potentially have an immunogenic response, because you don't have that to tell your body that this is yourself, so it's seen as foreign. And so this is really where we want to be cautious and not being able to expose a risk to patients as much as possible. But we're continuing to learn more every day about what is more or less immunogenic as well. Then there'll be more of the standard criteria for not being negative for AAV antibodies, and we will allow for washout to occur in patients if you have been on some of these other medications like givinostat or vamorolone in the past, but at the point of coming into the study, we want to make sure that you're not on those so that we can reduce variability. And as I mentioned, we'll be looking at this at the day 540, that 18-month time point, for a time to rise from supine velocity. So again, a time function test rather than something like the North Star ambulatory assessment, because we believe that this could be a more sensitive assessment. So again, learning from the field while also including additional assessments of ambulatory function so that we could really characterize what is this drug doing overall. And that really just goes to show what will that look like for a patient. And so someone who comes through the study, by the point of day 540, they will have the ability to get an active treatment if they had received placebo. And so really wanting to get all of the patients enrolled in the study on the drug at some point, either at the beginning or in the middle. And so with that, I really appreciate, again, everyone's attention here and sticking around for the end of the meeting. These are the QR codes for each of our studies to be able to get more information on the sites, on who's participating, on details. Inspire Duchenne, again, phase 1/2 that's active. Impact Duchenne that's upcoming soon. Both have clinicaltrial.gov postings that we'll continue to update as we have more information. But thank you all so much, and a huge thanks to all of the families, especially who are participating in this study. It's impossible to do any of this without you and all the sacrifice that you give, so really appreciate that, and appreciate all the investigative teams and the community overall, and Defeat Duchenne Canada. Thank you all.